So we are going to be presenting over this research article, Continuous Base Identification for Single Molecule Nanopore DNA Sequencing. So now we're going to move on to our introduction.
Sequencing has always been a valuable tool for biologists, used for things such as characterizing novel organisms or understanding organisms that we already know more accurately, such as using sequencing on humans for targeted therapeutics. However, it's had some huge problems, such as high cost, low throughput, and read errors. And this is mostly due to the polymerase used in current sequencing methods, such as Sanger sequencing or next-gen sequencing. This has made the Human Genome Project take way too long and be way too expensive because while we can get a lot of the genome with a constellation of contigs, it's hard to close it out, aka bridge the gaps between the contigs. And we hope that nanopore sequencing can solve this problem.
To preface the experiment, nanopore technology offers a potential for low-cost and high-efficiency sequencing without the need for fluorescence labeling with the use of exonuclease to free individual nucleotides. However, three problems arise from this method that needs to be addressed. First, the cyclodextrin adapters are not covalently attached, so there will be periods where no base readings are detected. Second, the resolutions of the sequence spaces are too low under the operating conditions. Third, the operating conditions were suboptimal for exonuclease activities.
Yeah, so that transitions us into how those listed problems will be resolved. Essentially, our hypothesis highlights how the use of a nanopore with a covalently attached adapter is able to enhance DNA sequencing in the sense that it's able to continuously identify unlabeled nucleosides. And, with a single molecule method like this one, cost efficiency and sequencing speed could also be improved upon. And so now moving on, we're going to tell you about how this was methodically carried out.
Yeah, so these are some of the methods that were utilized in this experiment. First, we're going to talk about how the team obtained the chemical reagents and enzymes. These were just bought from some established scientific suppliers, such as Avanti Polar Lipids, Sigma Aldrich, and Fisher Scientific. As for how the cysteine mutants were formed or created, the heteroheptamers were created by adding a sequence encoding a C-terminal tail of 8 aspartate residues to the genes of the cysteine mutants. And this was done through PCR mutagenesis and ligate-free in vitro recombination. Next, as for the setup, setting up the cis and trans side with a lipid bilayer, this was done by forming the bilayer of 1,2-diphentanol SM-glycerol 3 phosphocholine to a Teflon film, which divided a planar chamber into cis and trans side. And after that, we were able to add the salt solutions to both sides and also add the reagents needed for the experiment to both sides. As for the data recordings, this was made with a patch clamp, which zeroed out the current so that we were able to read the differences as the nucleotides were passing through the nanopore. And this was paired with in-house software that was able to collect that data. And once the data was collected, it was analyzed using standard methods such as the t-score.
So this is the critical experiment in the paper. Part A shows the electrical current passing through the nanopore as a function of time. So you can see that most of the current is at 50 picoamps, but it drops down to around 20 or 30. And this is when the nucleosides are passing through the nanopore. So this is the first experiment that shows that the nanopore can actually characterize nucleosides as they pass through, which is groundbreaking and leads the way for possible continuous strand sequencing using this method. However, one stipulation is this was not done with an actual DNA strand. It was fed free nucleosides and randomly they passed through using current. And this was done with 180 millivolts and 400 millimolar KCl. And at this concentration, it approaches 100% confidence in its nucleotide detection. And part B of this figure shows the event counts based on the current that it hit.
So now we're going to focus on the adapters, why those adapters are important, how those adapters were attached, and how the current disruptions during the sequencing were impacted. So a few things to note for figure A. In the transient adapter, open pore is when the adapter is not correctly attached, and this poses a problem because this renders the nanopore inactive and doesn't allow strands to read through, and thus no DNTPs will be observed. And building off of this, you can see when cyclodextrin is attached, the current is reduced from about 150 picoamps to 80 picoamps. And then on top of that, it must stay attached in order for bases to be read at all. For figure A, the use of transient adapters will, generally be less efficient when it comes to base readings. And so that's kind of why permanent adapters are objectively better. When it is covalently bound, it eliminates the open pore and inactive state. It's also important to acknowledge that in figure B, there are time periods when bases are still not being read. However, this is not because the nanopore was inactive, but because there isn't going to be, like a back-to-back flow of free nucleotides into the nanopore.
So to expand on the process of the bases passing through the adapter, the figure on the left here summarizes the average dwell time and the off-rate constant for each base. Figure A shows the average dwell times for each basis under standard conditions, and it is clear that the DTMP has a longer dwell time compared to the other three nucleotides. And this is probably due to the methyl group on the thymine base interacting with the adapter. And figure B shows the change in K-off over change in applied potential of the four bases. K-off is the off constant rate, and that indicates the rate of release from the adapter. If the DNMP is leaving through the cis side, a decrease in K-off is expected for increasing potential, since more potential promotes binding to the adapter. But if the DNMP is exiting through the trans side, the reverse is expected, since greater potential causes the base to be pushed through the adapter. So for DGMP and DAMP, K-off continuously increases, indicating that they exit through the trend side. For DCMP, K-off remains invariant. But on the other hand, DTMP shows a minimum at 120 millivolts, meaning that they return to the cis chamber at potentials lower than 120 millivolts and exits through the trend side at potentials higher than 120 millivolts. This means that a potential greater than 120 millivolts is needed to ensure that the nucleotides leave through the trans side instead of returning through the cis side and causing a possibility for them to be reread.
Now, in relation to the optimal applied potential per each nucleotide, the researchers are able to run that potential to ensure that the nucleotide travels from the cis to the trans side of the nanopore. By this, it is possible to measure the pore current when a nucleotide passes or discriminates, or they can identify the sequence between the four nucleotides. And that's exactly what the researchers have done here. In this figure, we are given two histograms plotting residual pore currents and the presence of these nucleotides. The x-axis is the current levels measured by the nanopore system, and the y-axis is the total count of each observed nucleotide. The researchers' purpose for doing this was to explore if the nanopore is able to distinguish between a methylated cytosine. Each peak in the histogram corresponds to a certain nucleotide run through the pore, where different nucleotides block the nanopore at different current intervals. Thus, the researchers can calibrate which nucleotides passes at a certain point. Now, the height of these peaks correspond to the frequency by which a specific nucleotide enters through the nanopore. And after running the first phase of the experiment, as we can see in figure A, they added a methylated cytosine in the second phase, to which it is clear that they are able to produce a distinguishable peak and therefore identify that this nanopore is able to identify the methylated cytosine as opposed to the other four nucleotides. This is important because it allows another layer of function for nanopore sequencing by its ability to detect these methylated cytosines. It plays a critical role in silencing certain genes, especially in the X chromosome inactivation, for example. Now to transition into data analysis and to recap the results and the purpose of the experimental procedures, the nanopore system was able to process and fairly accurately count for the 4 nucleotides. They're able to infer that the nanopore sequencing with a covalently attached adapter was able to discriminate between the 4 nucleotides and even distinguish between the methylated cytosine. This article found that the optimal salt concentration and the potential for the nanopore is most active under these conditions of 200 millimolar potassium chloride to 500 millimolar potassium chloride at 180 millivolts. Exonuclease enzyme performs most optimally at this as well, where the cis side is where the strands of the DNA will flow towards positively because the DNA is negatively charged. Now, this figure here incorrectly labels the optimal conditions at 160 millivolts, where the article indicates 180 millivolts. Now, in the supplement article, the researchers have provided a figure of the distribution of the 4 nucleotides across the optimal conditions with the corresponding confidence intervals for that base identity. Now, because of this, we can infer that nanopore sequencing technology is capable of sequencing some radically complex functions. This article gives no indication of a specific time interval by which to compare the relative speed to other forms of nucleotide sequencing. However, we can estimate based on the time interval averages from the figure 3a that was presented earlier, that the nanopore sequencing averages about 27 base pairs per second, or 1 every 0.037 seconds. And in a short research, nanopore sequencing is bested only by Illumina sequencing and relative speed. And this could lead to further research on speed efficiency and could have a market impact if improved. So in theory, this article's methods and research support the relative hypothesis that we gave by laying the groundwork by which a developmental method could be improved upon to use at a higher class or lab or clinical setting. This article does strongly support the discrimination of nucleotides, especially that of free-label nucleotides, through a covalently attached adapter to the nanopore, thus relatively supporting our hypothesis and transferring us over to the conclusions.
Okay, so now we're going to go over the conclusion. So here we see that researchers were successful in performing an unlabeled continuous sequencing with over 99% confidence, utilizing an engineered nanopore that has a covalently attached adapter. Some integration requirements to note were that the exonuclease must be close enough to the nanopore sensor, and the exonuclease active site exit trajectory must align with the nanopore entrance. Furthermore, the exonuclease rate may require manipulation to prevent misreads. So significant advancements in nanopore DNA sequencing is also supported by the research in this article. Overall, the research represents improvements in sequencing technology and addressing key challenges, as well as demonstrating the feasibility of a label-free continuous base detection system. The covalently attached adapter and optimized conditions enabled high confidence discrimination of nucleotides, paving the way for a nanopore sequencer. The potential applications of this technology are emphasized below, such as single molecule sequencing and epigenetics use. And now we're going to continue on to our final thoughts.
So we're going to wrap up this presentation with a next outstanding question. The question we wanted to ask was, what are the key technical advancements to enhance nanopore nucleotide detection that would allow for accurate and efficient continuous nanopore sequencing? This question builds off the success of this experiment in differentiating between different nucleotides as it passes through the nanopore. And we feel like the next steps would be to figure out a way to continuously sequence a long polymer or protein chain without the need of an exonuclease to individually liberate each nucleotide. Yeah, that wraps up our presentation.